All right, I am pleased to introduce Nicole Pagane, who did her practicum at Los Alamos National Laboratory and is studying systems immunology at the Massachusetts Institute of Technology.
Thank you. Okay, thank you very much. So I'm going to talk to you today about tissue-specific control of immune responses. So what is the immune system? The immune system is essentially a classification algorithm between self and non-self. And self here is any protein that is derived from your host genome, and non-self are any other proteins that could potentially exist that could harm you. And so how is this decision made manifest? Well, it's made manifest by how the immune system responds. And so the immune system is calibrated to not respond to self and to respond very dramatically to non-self. And that is, of course, to protect you. But in the destruction of potential pathogens, I don't know if this is working, but that's okay. In the destruction of potential pathogens, you could potentially destroy your own tissues. And so this input-output response curve is thought to be highly nonlinear to basically give rise to a very controlled immune system where you're not responding or you are responding. But the issue with such a digital classification scheme is, of course, edge cases, which the immune system can be vulnerable to. And so you have things like tumors that are mostly self with maybe a few key driver mutations that are non-self. You have the fetus, which is half maternal self, half paternal self, or you have commensal bacteria, which are entirely non-self. And now with this extended definition of self versus non-self, if you accidentally respond to functional self, you of course will get autoimmunity, but you can also have more miscarriages, or you would lack commensal bacteria in all of your surface barrier sites. And if you fail to respond to non-self, of course you would have persistent infections, but we would have much higher rates of cancers. And so the immune system must be flexible in this distinction between self and non-self. And so in maybe some cases, you want to have the flexibility to make this boundary more linear, or you want to set the actual activation threshold higher or lower. And the fact that the immune system must be flexible means it must also be controllable. And to understand how we can control the immune system, we have to look at the components of the immune system. And if you look at an immune network like this, it is highly complicated with many different molecular and cellular mediators, but I decide to study one of the most highly central high-degree nodes of this immune network called the helper T cell. And this hub essentially helps coordinate all aspects of immunity. So if you've heard of B cells and antibodies, T cells help with that. If you've heard of killer cells, these helper T cells also help with that. And so to understand how helper T cells exert control on the immune system, we have to understand how they're activated and how they're regulated. Dendritic cells present proteins to these T cells, and like I said before, they're pretty good at knowing if that protein comes from self or non-self, but that's not enough context to really understand if that T cell should respond. And so there are additional signals that the dendritic cell will also display, and there are diffusible signals in the environment in which these cells interact that can also influence that decision. There is a twin to these helper T cells called regulatory T cells, or Tregs, that use the exact same activating signals to become activated and exert immunosuppression. And so these Tregs essentially act as a sink for these activating signals for the T cells and can further lead to dampening of immunity. We can abstract a lot of nuances from these three cell interactions into a simple diagram like this, where you have dendritic cells directly activating T cells and directly or indirectly through a feed-forward loop activating Tregs, and Tregs exerting feedback control on the T cells. And so although this is simple, this could be a very powerful intuitive framework to understand how nonlinearities inherent to this circuit can prescribe a flexible boundary between self and non-self. Since we know in dynamical systems theories, very simple changes in very small, subtle changes in parameters of nonlinear circuits can give rise to very different functional outcomes. And so I'm really interested in understanding this module of control of how very subtle changes and differences between how these three cells interact can give rise to this functional, emergent, and dynamic boundary between self and non-self. And so if I want to study how subtle changes lead to potential different functional outcomes, I should study the circuit in its native environment. And this circuit exists all over your body in organs called lymph nodes. And lymph nodes are a distributed network of immune reaction centers that are strategically localized in different parts of your body to drain different tissues. And you can think of this as almost like a closed control feedback loop where the tissue will try to release signals to change the parameterization of the circuit, and then there's this reinforcement process where the output of that circuit, which is the T cell destructive power, will feed up into the tissue and then reinforce what is best for that tissue. But we have a lot of different tissues, and these tissues have very different functions: regenerative capacity, selection pressures, host critical functions. And so this trade-off of how to balance immunity to destroy pathogens, to recognize non-self, but to also maintain the integrity of that tissue, so to protect self, is probably balanced differently between these tissues. And so to study this, I look at lymph nodes that drain a bunch of different tissues in their native environment, so through imaging. And I use this really complicated iterative staining technique where I can look at 25 fluorescent protein targets at a time in a given lymph node. So here's a lymph node that drains the kidney. And we can look at, in green to the right, we see regulatory T cells that are activated and surrounded by different T cells, dendritic cells, and we can not just identify those cells, but we can also characterize their activation potential and regulatory status. And so then we get images, like beautiful images, if I say so myself, and then we can segment them to get statistics and to understand how the circuit parameters of these three cells and their interactions are different between different lymph nodes. And so I'm glad Ishani mentioned segmentation algorithms before. I do that to basically get something like this. And so for the analysis that I'll mostly talk about today, what I do is I look at the activated T cells in each of my lymph node sections, and I find the nearby regulatory T cells and the dendritic cells. And that's on the order of like 50,000 cells per lymph node. I have 11 lymph nodes per mouse. I have 6 experimental replicates of all of these images, and that ends up being 5 terabytes of raw imaging data. And so, of course, I am very lucky to use Mass General Brigham's ARIS computing cluster for most of my processing and analysis techniques, where I'm just constantly requesting hundreds of nodes to just run my analysis in parallel so that I can immediately start to make some impressions of my data. Okay, so how are these circuit parameters different between different lymph nodes? In the purple graph, or the purple bars, we see that the dendritic cells seem to carry the most variance between the lymph nodes, where then there's this hierarchical level of next level of regulation, where the regulatory cells in the gold bars have the next most variable features between lymph nodes, and then the conventional T cells in green have the least. And so this started to tell me that maybe there's information from the tissue that is encoded hierarchically and fed forward through this feedback circuit so that you impart tissue-specific characterizations. And since dendritic cells seems to be important to understand the tissue-specific differences, I've decided to try to further classify those dendritic cells, since dendritic cells have been characterized into different functional subclasses. And immunologists love to gate cells and say this cell is this type and that cell is this type by just looking at 1 or 2 markers and setting a threshold. I don't like that approach because, as you can see, dendritic cells are very irregularly shaped, they're dense, they have connecting cell bodies, and so I decided to build my own ensemble machine learning classifier where I generated my own labeled trainings data set, I'm happy to talk about it if you're interested, so that I can actually classify all the different features on the dendritic cells to have a more accurate, with uncertainty metrics, values for these classifications. So with this machine learning ensemble classifier applied to my data set, we start to see that the subset proportionals are very different between lymph nodes. Don't worry about what the different subsets do, it doesn't matter. But the orange bars are thought to be more baseline inflammatory, and the green lines are thought to be more baseline tolerating. And so we have our first inkling of maybe like the lung and brain lymph nodes seem to be slightly more inflammatory, which I thought was weird at first, but a lot of autoimmune diseases and immunopathologies happen in the brain and lung. So maybe there is some weird setting that way, where the liver and the gut draining lymph nodes seem to be baseline more tolerogenic, and then something like sex draining and skin lymph nodes are somewhere in the middle. But we have so much more data than just the dendritic cell subsets. So I do what every big data person does, and so I take my larger dimensional space, project it into a lower dimensional embedding through PCA, and I start to make insights that way. What immediately struck out to me is that the shape that constrains my data is a triangle. And I find this interesting because I've posed this problem essentially as a multi-objective optimization problem, and there's a really cool trick in Pareto optimality that can help make sense of the shape. I'm sure most of you know what Pareto optimality is, but in case you don't, a really simple example is to just look at people in this room. We have a lot of computer scientists that are probably really good at computer science. We have a lot of domain specialists that are really good at their domain. And then we have interdisciplinary scientists that are not as good at one of those two tasks, so they're specific domain or computer scientists as those specialists, but they're pretty dang good at it. And so the idea with multi-objective optimization in the Pareto front is that there's a manifold of solutions that are all optimal depending on what you want to prioritize. You won't exist below the front because you wouldn't be in this room because you wouldn't be interested in what you're doing, and you wouldn't exist above the front because you can't specialize in two things at once, and if you can, please tell me where you find the time and energy. But anyway, the theory goes that these extremal specialists exist in trait space as extremal archetypes. And all of the interdisciplinary generalists are some type of linear interpolation of those archetype features. And so if you have 2 tasks, you get a line, 3 tasks, a triangle, 4 tasks, a tetrahedron. And what's interesting is that there are decades of ecological research that suggests that different species that live in different ecosystems occupy lines, triangles, and tetrahedrons. And their effects or their features are associated with what people assume they eat or what they use to adjust to their climate. And so here we can try to label the vertices. So I have an inflammatory axis where the T cells, you can't see the arrows, but the T cells are upregulated in the top vertex, the anti-inflammatory axis where the Tregs are really activated, and the far vertex is where the DCs are activated, but not so much the other ones. And we can start to paint the points by what tissues they come from. And so the lung seems to be slightly more pro-inflammatory. The gut seems to be anti-inflammatory. The skin seems to be this weird poised but not activated state. And something that drains the sex organs is a generalist instead of a specialist. And while that is a really powerful framework, it is correlative at best. And so what I'm working on is using multi-objective optimization where I can jointly model through ODEs tissue features and the actual 3-cell circuit in lymph nodes. And then from the data, actually inspired from the data, write explicit objective functions that of course are going to be some combination of our state variables from our dynamical system, and then run multi-objective optimization to find the solution set of parameters that can balance these different objectives. I use the deep Python package, which is uses a bunch of evolutionary algorithms that mimic evolution processes like mutation and mating and selection pressures to evolve your parameters over time. We can end up with this in silico Pareto front of our tissues, and we can start to see how these different parameter instantiations give rise to different dynamics, and we can actually do experiments in the lab to validate some of these predictions. I haven't done those experiments yet, but what I can tell you is that if you look at the solutions, or the parameters sets for the different solutions on the Pareto front, we see that tissue properties seem to drive a lot of the variation, but really the DCs are the predominant parameters that are different, which is reflective of my IBEC or my imaging data, where we saw most of the differences were in the DCs, and then also some in the Tregs. So at least correlatively, we have a new way to validate that the archetypes we're inferring and the tasks we're getting at might be something real. And so in summary, I hope I've impressed upon you that this is a really important circuit to study immune regulation and control. Immune regulation and control is really important for setting this dynamic boundary between self and non-self, and that different tissues likely handle these, prioritize these tasks differently. And I want to thank the lab, of course. I want to thank other collaborators I've had on other projects that have really helped me become more of an immunologist. And of course, I want to thank my practicum advisor, Alan Perelson, who basically created the field of systems and quantitative immunology and inspired me to get into the field in the first place. So thank you.